Laboratory techniques for genotyping and sequencing in pharmacogenomics. Genotyping and sequencing are fundamental laboratory techniques used in pharmacogenomics to identify genetic variations that influence drug response. These techniques allow researchers and clinicians to analyze an individual's genetic profile and determine the presence of specific genetic markers associated with drug metabolism, drug targets, and treatment outcomes. Here are some common laboratory techniques used in genotyping and sequencing for pharmacogenomics. 1. Polymerase chain reaction, or PCR. PCR is a widely used technique in molecular biology and pharmacogenomics. It allows for the amplification of specific regions of DNA, making it possible to detect genetic variations in target genes. PCR involves cycles of DNA denaturation, primer annealing, and DNA synthesis using a DNA polymerase enzyme. PCR can be used to amplify specific regions of interest before genotyping or sequencing. 2. Sanger sequencing. Sanger sequencing, also known as chain termination sequencing, is a classic DNA sequencing method. It involves DNA amplification by PCR, followed by incorporation of chain terminating nucleotides during DNA synthesis. The resulting fragments are separated by size using gel electrophoresis, and the DNA sequence is determined based on the order of terminated fragments. Sanger sequencing is suitable for analyzing individual genes or specific genomic regions. Next generation sequencing, or NGS, technologies have revolutionized genomic research and pharmacogenomics by enabling high throughput sequencing of DNA. NGS allows for the simultaneous sequencing of millions of DNA fragments, providing comprehensive genetic information. Different NGS platforms, such as Illumina Ion Torrent and PacBio, offer various sequencing approaches and applications. NGS can be used to sequence entire genomes, exomes, targeted gene panels, or RNA for pharmacogenomic studies. Microarray technology uses a grid of DNA probes to detect specific genetic variants across the genome. The DNA samples are hybridized to the microarray, and the fluorescence intensity is measured to determine the presence or absence of specific genetic markers. Microarray genotyping can analyze a large number of genetic variants simultaneously, making it efficient for pharmacogenomic studies that focus on predefined genetic markers. Mass spectrometry. Mass spectrometry based genotyping methods utilize the principles of mass spectrometry to identify and quantify specific genetic variations. Techniques such as matrix assisted laser desorption ionization time of flight, also known as MALDI-TOF, and selected reaction monitoring, or SRM, allow for the high throughput analysis of genetic variants. Mass spectrometry-based genotyping is often used for single nucleotide polymorphism, or SNP, genotyping and detection of specific genetic variations. Digital PCR. Digital PCR is a sensitive technique used for absolute quantification of DNA molecules. It partitions a DNA sample into thousands of individual reactions, allowing the detection and counting of specific genetic variants. Digital PCR is particularly useful for detecting rare genetic variants and accurately determining their abundance in a sample. DNA microarrays for gene expression analysis. Gene expression microarrays measure the levels of gene expression for thousands of genes simultaneously. They allow researchers to study how genetic variations influence gene expression patterns and identify biomarkers associated with drug response. Gene expression microarrays can help uncover gene expression signatures relevant to pharmacogenomics and personalized medicine. These laboratory techniques enable researchers and healthcare professionals to identify and analyze genetic variations relevant to drug response. They provide valuable information for individualizing drug therapy and making informed treatment decisions based on an individual's genetic profile. The choice of technique depends on the specific research or clinical question, the number of genetic markers of interest, and the available resources and infrastructure.